Thank you, Dmitry. And so, yeah, some time ago, I was working on a virtual screen project. And it is where you train some machine learning model to predict molecular properties, and then you apply it to a huge list of molecules that you can order. And then you select the best ranked molecules and test them in a real life experiment. So there was a point where baselines were done, so we wanted to do something more fancy. We wanted to find maybe some state-of-the-art models, pre-train them, train them, stack them, something cool. So I sat down and started reading papers. And I was reading them and studying. But the more I read them, the more confused I was. At some point, eventually, honestly, I couldn't tell if machine learning is even work on novel molecules rather than memorize training data. So while I was trying to answer my questions, it led me to a new benchmark, a new library, and eventually a paper, low-high practical machine learning drug discovery benchmark, which was published at NeurIPS 2023. And that is what I'm going to tell you about. I'm Simon Steshin, deep learning researcher. So, as I said, I couldn't tell if machine learning works on novel molecules, but now I know, and I will tell you. And today we'll learn if it works on novel molecules, and can a machine learning model distinguish minor modifications in molecules. And it is not a simple question, because if you know something about those models, you know that you can train them on binary fingerprints, but those are very discrete representation, because different molecules can correspond to the same binary fingerprint. Or you can train graph neural networks, which is limited by which has limited expressivity, meaning that different molecules can be mapped into the same embedding. So can they distinguish minor modification? It is really important if you want to use it in the optimization process. So what if you have some hits, some molecules you really like, but it is not quite there yet. So we would like to improve certain properties of it. For example, penetration of blood-brain barrier while still being active. So then I will tell you which models are better for searching of novel molecules, which are better for optimization. And what is really interesting, that is, those are different models. That is interesting for me. I will tell you what's wrong with modern benchmarks, and then I'll tell you how does low-high benchmarks solve at least some of these problems. I don't really expect that you know anything about chemoinformatics. I expect that you know about what is the machine learning, or at least maybe what molecules are. I'll try to keep it shallow, not too technical. Okay, so the first thing I need to say is like prerequisite, and it is molecular similarity measures. So imagine it is a molecule, it is a real drug. You can go to pharmacy and buy it, and it is a drug for a certain disease. And then I'll come to you, show you this molecule, and I say, look, I found a novel drug. How cool is that? Well, you won't be really impressed, right? It is almost the same molecule. Not cool. I cannot patent it, right? What if I bring to you this molecule? And this is another case. It looks very dissimilar, right? So how can we tell which molecule are too similar, which are dissimilar, which are novel? And first, how can we measure similarity between them? And it is I think it is like the main question of chemoinformatics, molecular similarity. It was studied for at least 20 years, but I would say probably 50 years rather. And there were many papers about them. So there were many papers that said that, well, we have a lot of different ways to measure similarities between molecules, but which is the best? And then they formulate some desiderata, right? So they formulate which measures should be considered as the best. And then they test different measures and find the best one. And apparently, all of them find one molecular similarity measure as the best, and it is Tanimoto similarity on ESFP4 fingerprints. If you don't know what is it, it's not important. But what is important, that this is the gold standard. It is how most people think about this. That is what they use. So we will stick to that. Okay. You can see that if a molecule really looks like a known drug, like if two molecules are really similar, then the Tanimoto similarity will be about 1. And if they are really dissimilar, it will be about 0. So can we think about some threshold? So molecule that is more similar than a threshold be considered as too similar. Is it possible? And honest answer is no. It is subjective. If you faced with some of such problems, you need to think about your goals and formulate your understanding of novelty. But here are some ways how we can formulate this. First, we can think of patentability. We can call a molecule novel if we can patent it. It is cool, but it is not practical, because it's really a daunting task to determine if a molecule can be patentable. There are companies that can do that service for you for money. And the reason it is not automated yet at this moment. And the reason why is it so? Because patents are written in a very difficult way. It is you cannot automate it yet. There is no such databases. So you cannot test your chemical library with 1 billion molecules for patentability. What could you do else? One could think of, one could hope to find some thresholds of similar properties. So it would be great to find a threshold and say that if two molecules have a similarity larger than some threshold, then they likely to possess the same properties. However, it's not work, because for different properties you will find different thresholds, right? So it was said that the similarity is in the eye of the beholder. And there is an interesting subfield on the intersection between psychology and medical chemistry. And there are work that tries to disentangle and formalize intuition of medical chemists. For example, you can bring to medical chemists different molecules and ask them which molecules they like more, which they would like to prioritize as lead development. And apparently different chemists give different answer. They give different explanation for them. And even if you bring them the same molecules again after some times, they will choose different molecules for different reasons, which is fascinating. And one of those kind of one of those kind of papers tried to measure and formalize a notion of similarity. It is a work from 2014. And if you developing a drug for orphan disease, you can get an orphan drug designation, which is a regulator can issue it, and it gives you some benefits. And for example, EMA, European regulator, if you look into its documents, it doesn't really formalize idea of novelty. but the procedure of determining the nullity is basically majority voting. So you ask experts, and if more than a half expert think that your molecule is novel, then it is novel. So this paper tried to formalize that intuition. They took 143 experts from mostly regulators like FDA, EMA, FDA of Taiwan, etc. And they showed them 100 pairs of molecules and asked them if they are the same molecules or not. They also tried to predict that intuition using different fingerprints. You can see BCI, daylight, of course, ESFP4. And here is the result. On the x-axis, you can see Tanimoto similarity. On the y-axis, a fraction of expert that thought that the pair was too similar. So you can see that half of the experts would say that your molecules are dissimilar at 0.4 Tanimoto similarity. It's like a threshold, right? So as a rule of thumb, you can choose this threshold. There are some drawbacks like size bias, but anyway, Tanimoto similarity is fast, it is cheap, simple, and actually there are theoretical and practical work that use actually 0.4 Tanimoto similarity as threshold. So we will stick to this value. So in this paper, we will call molecules novel if they are less similar than 0.4. Okay, so let's get back to the paper. Hit identification. It is the first task. And what hit identification is? It is an early stage drug discovery. It is early stage of drug discovery pipeline. Imagine you have some disease and you probably know something about its mechanism. You have some ideas about, you select your target that you would like to maybe inhibit. So at hit identification, you would like to find any molecule that do the task that bind and maybe inhibit your target. One way to do that is high throughput screening. When you buy like 100,000 of molecules and you run massive parallel experiments of all of them. And another way to do that is virtual screening. So suppose you have a data set of activity of some molecules on your target and you train machine learning model on them, and then you make prediction of that model right on your computer for a huge list of molecules that you can buy at some shop online, right? Then you select the best molecules and send them into real life experiments. It could be cheaper, right, than higher throughput experiments. In practice, there is interesting property. If you train your machine learning models and you apply them to rank a huge list of molecules, it is very probable that your model was slightly overfitted on your train set. So at the best ranked molecules, you will find almost the same molecules that were in your train set. It is because your model seen them, it is very confident that those molecules are active. And it is not what you're looking for, because it is quite trivial to find that very similar molecule work in the same way. You would like to find a novel molecule that is maybe patentable. So here is a plot depicting that. So imagine you have a train set and where you train your model, and you're going to apply it to molecules that are very different from your train set. This is not always true, but sometimes, yeah, people do that. So what you can do is you can take your chemical library, a list that you're going to make prediction of molecules in that list, and you just filter out anything that is too similar to your train set. You can filter those molecules by Tanimoto similarity, and that is what's done in those 6 papers. You can see those are good prestigious journals with some famous scientists. So it is quite a standard procedure, right? However, when I was reading those papers, and why I was interested in that, because I wanted to do the same. I would like to apply my model to predict, to find novel molecules, right? When I was reading those papers, I found interesting discrepancy between how models are trained and selected and how they are applied in practice. So the difference is that the authors select models that works best on molecules that are really similar to the train set, but apply them to the molecules that are very dissimilar to the train set. It could be not optimal. So why is it so? Let's look at how they split their data sets. And there are basically two ways of splitting it. And you probably understand random splits. Random splits, if you have a data set, you can randomly split it into train and test. However, it's very probable that you will, that there will be some molecules in the test set that are very similar to the train. As it said here, random splitting common in machine learning is often not correct for chemical data. And as we are more interested in discovering new categories of, for example, HIV inhibitors, scaffold splitting is recommended. 2018. So what scaffold split is? There is such thing as scaffold. It's a paper from 1996 by Bemis and Murko. And scaffold is like a simplified representation of molecule. You can see here on the right is a scaffold, and this molecule on the left. And sometimes different molecules can be mapped into the same simplified representation, in which case they all goes into the same split, either all to the train, either all to the test. So it's kind of should generalize better, right? Let's look how it works in real life. So here is ESOL. ESOL is a very famous benchmark in machine learning on chemical data. It's very popular. It's very ubiquitous. And here is ESOL that was splitted by canonical random split, as it's always splitted. And it is, I calculated a new distance between test and train molecules. As you can see, most of the molecules in the test set have a pair molecule in the train that is very similar to it, right? Here is 0.4, our threshold of similarity. What is even fascinating is this peak. It's 1.0, meaning that there are 4 molecules in the test set that has the exact clone in the training set. Interesting. So let's look at scaffold split. Maybe it will work better, right? No, here is HIV dataset. It is very common as well. It's a phenotypic screening of inhibition of HIV. It has 40K molecules, which is great. And you can see that most of them have a very similar molecule in the test set, in the train set. So it is probably not good if you are going to apply your model to really novel molecules, right? That is a discrepancy. If you're going, like those 6 papers, apply virtual screening on novel molecules, you need to select model that works best on novel molecules, that generalize the best. That is what I think really good thing to do. And it is not done yet. So that was about hit identification. So I would formulate hit identification task as testing generalizability, right? So in hit identification task, you have train and test that are very dissimilar from each other. And you would like to find models that work the best on dissimilar molecule. That is hit identification or high. Okay. And let's go to the second task, which is lead optimization or LO. And lead optimization is usually the next step in drug discovery pipeline. So imagine you found some cool hits, you would like to optimize it. As I said, you can improve some, for example, penetrability of blood-brain barrier, or maybe improve its stability or anything like that. And So it is very, very related to goal-directed optimization, which is task machine learning task in generative models. So it is formulated in different ways. For example, you can formulate this task as generation of the molecule's maximal activity within the epsilon neighborhood of a known hit, or maybe not epsilon neighborhood, but of a known scaffold. Or maybe you'd like to optimize something in the latent space. It is a whole subfield of molecular generation. It is well known. And for example, you might hear about this paper even if you never studied anything about cheminformatics. It is a seminal paper from 2017 by Gomez Bombarelli. What they done, very simple. They trained variational autoencoder that encodes a molecule into latent space and decode it from latent space. What is interesting that they also trained a property prediction from the latent space, right? So it is a property of interest, maybe some chemical property, right? What you can do now, you can take a molecule, encode it into latent space, and move it here to maximize predicted property, right? Then when you found the best optimized molecule, you can decode it back to the real molecule, which is cool. So how does it work in real life? Here is an example. It is a paper from 2022. It published in a very prestigious machine learning journal, Nature Machine Intelligence, and it is a group from big pharmaceutical company Novartis, which is cool. What they try to do, they basically done the same model, but it was variational autoencoder on graphs, and they try to optimize molecules for antimalaria's property, right? And if you read this paper, if you maybe don't read many papers in this field, or you read this paper quickly skimming through, it might look like a positive result, like a good paper with good findings, right? Our findings show that they model the viable approach for finding novel active compounds for drug discovery. Cool. However, when I read that paper, it looked not exactly that. The narrative has some holes in it, which could be connected and in an interesting way. And it seems to me that they have a negative rather result. They tried to do that. They tried to optimize molecules, but they couldn't find molecules better than they had. The optimization probably didn't work. Anyway, this is very cool paper because I really appreciate it, because from all the papers that use molecular generation, there are about 12 papers that try to test it in real life. And this is one of the bravest 12 papers, which is really cool, very important, but the results is rather negative. Why is it so? It is an important question. What could go wrong? And one of the things that could go wrong is this property prediction. You see, they run, it was, well, based on optimization algorithm, but basically it's hill climbing, right? And to do hill climbing, you need a convex landscape. It should be good, right? If your property prediction model is very noisy, if your predictions are noisy, then your landscape will be noisy as well. There won't be those hills, there would be peaks and holes, and you will get into a hole, you won't optimize anything at all. So what I would like to know is, could those machine learning models distinguish minor modification of molecules? Do they have those good convex landscape as depicted in this picture, or is it rather noisy thing that can predict activity on average but cannot distinguish minor modifications? Apparently nobody tested that. That was really interesting for me. So that's what I tried to test, and I tested that in benchmark that I called a low lead optimization. So the point is that, so imagine you have a train, and the test is represented as clusters of very similar molecules. The goal of model to rank those molecules, to say which molecules are better, which are worse. There are a few technical details. I omit them here. You can find them in paper. But few things. First, I take exactly one molecule from each cluster and put it into the train to imitate a known hit. I also use a Spearman correlation coefficient that is not sensitive to outliers. So it's just ranking from worst to the best. So it doesn't depend on variation within a cluster. And the third thing is that I ensured that the variation within a cluster exceed the experimental noise. It is important because it's meaningless to try to predict experimental noise, right? So that is the lead optimization task. And before I get to results, I will show you what worked, what not. I will tell you how to do it properly, how to do those splits, how to prepare those benchmarks. And as for the low splits, it's pretty straightforward. The code is complicated, but the idea is nothing fancy. Just code that down and everything will be fine. But for the high split, as I said, you would like to split your dataset into training and test such way that no molecule in the test set is similar to the training set. A greedy way to do that is to do split by any way, for example, random splitting or scaffold splitting, and then just remove very similar molecules from the test set. Just forget about them, right? However, that is not cool because you lose some data points. And it is important in cheminformatics where each data point costs you in order of $1,000, right? We already have small noisy data sets. We would like to minimize number of discarded molecules. So how to do that? So let's formalize the problem. Let's say we have a graph corresponding to our dataset, and each vertex corresponds to some molecule, as is depicted here, and two vertices are connected with an edge if their molecules are very similar to each other. Right? These two molecules are very similar. They are connected with edge. This is dissimilar. This is not connected. So you can see that you can take connectivity components and assign them to the training test independently, which is good. However, in practice, in real life, unfortunately, most of your data sets will look like that. In real life, you have probably 95% of your molecules in one giant connectivity component. It's not that dense as here. It is just to intimidate you. But in real life, it's like sausage, right? You cannot, it's not straightforward how to put it in train and test. So the formalized problem is that we have a connected graph. How can we split it into train and test in such a way that we minimize number of discarded vertices? And with respect to some constraints on the sizes of train and test, because we would like to have train has like 90% of molecules on the test, like 10%. And apparently this exact problem was studied in computer science for the last 20 years, and this is called minimal vertex K-cut. It is an NP-hard problem, but it is solved by linear programming, by some heuristics. And I won't delve into the math. And to be honest, I added those formulas just to impress you. But the math under that is not that difficult. It's actually very, very cool and simple, right? So if you are interested, go and read that. It's linear programming. One thing to note is that in those papers, people formulate this problem and they suggest some algorithm how to do it faster. However, the biggest graph that they use, I mean, in computer science papers, the biggest graph they used is like 300 vertices. And I have, like in case of HOV graph, 40K vertices. So when I implemented this formulation, it was just too long, way too long for real life datasets. So one way I done, I just, as a pre-processing step, I clusters molecules using Butina clustering. And so the graph was actually, I got a condensed graph, more coarse graph. So in my graph, each vertex actually corresponds to several similar molecules. And that trick helped me, and I done HEV dataset splitting in maybe several minutes. So as we can see, this result works really much better than the greedy approach. Because, for example, for DRD2 activity dataset, it's lost only 97 molecules instead of 1,000 molecules, which is cool. So that is how you can do high split. And luckily, if you don't want, you don't need to. I already made a library, just install it by pip install low-high splitter, and use it in one liner. So you don't need to know that's linear programming techniques. So let's get to the results. So do models work? Do they generalize? Do they sensitive enough to those minor modification? And apparently, yes, they are. So I prepared 7 datasets, 4 for high task and 3 for the low tasks. And they are very, there is a wide range of tasks. It is activity to GPCR receptor, to kinase receptor. It is phenotypic HEV dataset and solubility dataset. Yeah, and I also prepared folds. So it's not trained test, it's 3 folds. So I accounted for difference in data, and I done meticulous hyperparameter optimization for each of the model for each data set. It took some time, but the results worth it. So as we can see, yes, models can predict out of the trained set. It is far, far from perfect, but we can see that ChemProp, which is graph neural networks, works the best. It is really cool. It's really improved over dummy baseline, which produced that base rate that's one value. What is interesting that for the low tasks, the best model was support vector machine on ECFP for that fingerprints. So it was better than the graph neural network, which is very interesting. It is surprising for me. One could say that graph neural networks have limited expressivity, but fingerprints have limited expressivity as well. But it is on par with previous work on activity cliffs. They also found SVM to be better than neural networks, which is very interesting. I would say that the main outcome of this work is that different models are better suited for different tasks. So I would recommend you to think about your task and prepare your model selection pipeline appropriate. So thank you for watching. Join ComputAge chat by this QR link, and I'm happy to answer your question.
Simon, thank you very much. I really love these results, and I love this task because it's strongly in line with something I'm doing right now. Just I'm doing before I started watch this presentation by you. So guys, questions please.
Can I ask a question?
Sure, Marina. Hello.
Yes, thank you, Simon, for the presentation. I was a little bit late. Could I ask a question? Probably you already covered it, but well, I'm sorry. Could you please clarify what is the setup for the models that predict that identify hits and that predict that optimize the ligands? So, what is the input on what data they trained? And yes, basically, what is the setup for this task?
Yeah, for sure. So for hit identification, it is you should have a data set of molecules and a binary label. If it's active or not, does it possess some property of or not? That is hit identification, and it should be splitted in that way. So your test set is really dissimilar to the trained set. So you are testing for generalizability because you would like to find null hits. As for the lead optimization, it is I would like to test models for their ability to be sensitive to minor modification. So your data set is molecules and some continuous value. For example, solubility or maybe activity, yes, as a continuous value. And then you run LO splitter that will make those clusters. You train on train set and test it on clusters, and you will get a Spearman coefficients from 0 to 1.
So basically, there is no information about the protein because these are kind of applied data sets for a single protein, and we are just learning the properties of small molecules that target this protein, right?
Yeah, absolutely. In drug discovery, there are two approaches to some fields. It is ligand-based drug discovery and structure-based drug discovery. And in structure-based, as you said, you have some information about your protein, right? Maybe sequence or maybe structure itself. And I was talking about ligand-based drug discovery because in ligand-based drug discovery, there is no information about the protein that goes to the input. And it could be cool, for example, because not all the interesting properties are activity, right? Because you might be interested in solubility, you might be interested in some metabolic thing, in log P, in blood-brain penetration barrier, and anything like that. And that is ligand-based drug discovery, yes.
Thank you for clarification. And this in this table, these marks, EC, blah, blah, these are some models, right? Or is it?
Yeah, on the left. Yeah, those are models. At first, there are models trained on binary fingerprints, and down here to model that trained as graph neural networks on graphs.
Don't you know that anybody tried some fancy diffusion stuff or some kind of complicated architectures for ligand-based drug discovery? Because it's kind of, at least in structural bioinformatics of proteins, I'm so already not used to see something like KNN and the SWAM in the table of methods.
Yes, ChemProp and GraphFormer are both fancy methods. ChemProp is a message passing neural network that is directed. It's literally direct message passing neural network. It was introduced right with ChemProp, and GraphFormer was a paper from Microsoft. It was a mix. It was the first graph transformer that set to work, right? And I would call those as a fancy architecture. As for the simple architecture, I use them because they are just fast to train, fast to test. And there were interesting results as for activity cliff that supports vector machine. There are some papers that say that classic machine learning algorithm works better. And apparently, sometimes they are, right? Like here with LO task. That's what I think is important to establish strong baseline to beat it. And you said about structure-based and diffusion, but I guess you're probably talking about generative models, because diffusion is a generative model that generates, maybe you're talking about those papers that generates elegant for exact structure. But that was, you're talking about generative models, and this benchmark is about molecular property prediction. So it doesn't generate anything, it just predicts.
Yeah, I see. And what is the average size of data, these data sets? So how many ligands are there?
Yeah, I included a data set of different sizes just to, because of this would be interesting. So HEV is like, it is a huge data set of 40,000 molecules. Dopamine receptor and KDR are like 7,000 molecules, I think. Solubility, I think 500 molecules. It is a medium-sized data set, I would say.
And these classic molecular machine learning models, they what descriptors they use for ligands?
Yeah, you can see there are in brackets, they are either ESP4 or MAX. I tried 2 of them for each model.
Sir, could you characterize shortly these descriptors, what they include?
Oh,
it's
I
a kind
cannot.
It's a kind of just a feature of molecule. A number
Yeah.
of features. Yeah.
Kinda. As for the MAX fingerprints, I think they have a bunch of fragments, like 140, I think, something like that. And it's just, as you said, it's fingerprints representing if those exact fragments are within a molecule or not. And as for the ECFP4, it's not that. It's a kind of different type of fingerprints. It's not about fragments. It is, you can characterize it as a hash of a molecule. It's very interesting how it builds. It's a difficult procedure, but I would consider it some kind of hash of inner fragments, something like that.
Okay. And let me ask one more.
Sure.
Could you shortly clarify the term expressivity of a network?
Yeah, I can. Expressivity, I use that term when I was talking about graph neural networks. And expressivity, the problem with vanilla graph neural networks is, as I said, they cannot recognize some graphs. So if you build vanilla graph neural networks, you can find some graphs that are mathematically embedded to the same embedding all the time, independently of your training. It's just the way how it works. So it is the question about isomorphism of graphs. You know, it is an NP-hard problem. There are different heuristics to check to graphs for isomorphism. And vanilla graph neural networks doesn't solve that because they cannot differentiate some graphs, or in this case, some molecules. However, what is interesting, it is hot topic. I don't know it quite well, but there are always new and new architectures of graph neural networks that solve exactly that problem and tries to make models that are more expressive.
So I'm not sure I got the term of expressivity. Probably better I will read. I think
Okay.
I just missed something. Missed a particular. Just mislistened maybe
Sorry.
something. Do we have another participant, Sergey? Do you have a question? I can give you a turn. If not, then I would like to ask my last question.
Sure.
Maybe the most interesting for me now, because I really since recent time I constantly face with the problem of uncertainty in machine learning, especially with out of distribution uncertainty and how model estimate their out of distribution uncertainty in clinical context, in general machine learning context. For example, if we predict some molecule to be hit, to just the label 1 for a particular molecule, regardless the exact what we want to predict,
Yeah.
how can we be sure that this prediction is reliable? The modern frameworks of molecule predictions bother about this problem.
Yes, I would say that first, how you can be sure that that's a valid hit from like maybe Bayesian points of view, from decision theory. You can say that, well, my model worked at the test set quite good. It has good metrics. That is why that is in this amount of certain that my model will work for this exact molecule. But I guess you are asking about uncertainty estimation, which is a subfield in machine learning, right?
Yes.
Yes, and there are actually a bunch of works that tries to do uncertainty estimation for molecular models. Yeah. For example, this benchmark doesn't handle using any uncertainty, but yeah, there are many results. And for example, ChemProp has inner model that can predict your probabilities, right? But as I know, the problem that I faced when I was trying to do uncertainty estimation is that it's often needs additional data. You need data to calibrate maybe your models. And it is very sad since we already have like, what is it, 500 molecules here. It is already sad. And that is why I didn't use it. I use it never, but yeah, it is a field. People do that. Does it answer your question?
Yes, I think it is enough for answer. I see that some models actually bother about uncertainty estimation. The only thing I would to clarify is if we just suppose we face with the we trained our model somehow and we have a good metric with test set, but since some time we took additional molecule which is so far from our train and validation and test set we had before. For example, this molecule is purely out of distribution molecule.
Yeah.
In this case, can we be sure that model predict whether this molecule hit or not hit correctly? can we rely on the probability score which model returns us, or can this model estimate its level of uncertainty about the prediction? For example, Kim-prop model you mentioned. Can it estimate the uncertainty of its prediction for totally new molecule?
Can we be sure of prediction for auto-distribution molecule? Of course not. For many reasons. There are, when you study machine learning, there are always that notion of assumption of it's the same distribution, right? It's everywhere. So if you take that out, then you can't rely on anything. You will get no guarantees on anything. And that is very important question, because as I said, many people are interested in applying those molecules in some kind of auto distribution formats. For example, when you apply it for molecules that are filtered by TNMOS similarity to the training test, and to somehow imitate that auto distribution difference, I suggest to use high splitter, right, to imitate those discrepancies, and it will help you to choose more appropriate models for that. It is not that overfitted. As for Kim-prop, will it work on? Will it return low estimates for novel molecules? I don't know, but I think it very well could, because it was there is a very forgotten technique in molecular property prediction. It was ubiquitous before neural networks. It's called applicability domain. So when medical chemists trained machine learning models before, like 15 years ago, they always were talking about applicability domain. So they knew that they can apply those models only in certain very limited chemical space. So yes, they literally have included if in their code that check if the molecule entered that applicability domain, and if it isn't, the model just decline your query. But yes, it's if you want some generalizability, you want generalize with this approach.
Thank you for the answer.
Can I ask a question?
Sure.
Yeah, thank you. In line with previous question and answer, would your model work with bigger molecules like polyoligosaccharides, polyoligosaccharides? And the second part of the question, would your model work with amyloid-like structures? I mean, not just a single protein, but let's say 3, 4, more than one unit of protein. So oligomerized drug and oligomerized target, would it be compatible in your model? Or, well, workable, I don't know how to say it.
No, Sergey, it won't work. Because those models that I tested, they are created for only small molecules.
How small?
But like
so is it below 1,000?
Yeah, below 1,000.
Yeah, okay, all right.
However, there is a huge research on different modalities. You might heard about some model that trained on peptides, just sequences of peptides. There are some machine learning for antibodies, and of course, structure-based drug discovery that incorporates protein information. But I don't know much about that.
Okay, thank you.